Okay. Hello, everyone. Um, I know my background is rather strange, only because I am driving from Del Rio to San Antonio. I had the unfortunate experience of having to officiate a funeral, and so I won't be able to be in class, obviously. But so by the time you see this recording, it's because I've missed class. But I want y'all to watch this recording, and if you can, not if you can, I would like for you to write a three-paragraph, um, just a three-paragraph reflection on the video. As I mentioned, we're going through some uncharted waters as far as the neuroscience field, and as a result of that, I'm introducing you into something that may be somewhat unfamiliar to you, and I definitely taken that into consideration. But I assure you, as I have in the previous class, that it's going to benefit you greatly when y'all become clinicians. You will be able to communicate effectively with psychiatrists and other medical providers, as well as understand a little bit more in greater or understand in greater depth the psychopharmacology that your clients will always present themselves with in their history or their in your assessments. So, with that said, I'm going to go ahead and get to my notes here because I want to go over chapter two, um, the cell transport. What I mean by cell transport, and of course, the video reflection I'm going to include on there so y'all can so to further solidify and strengthen your understanding of what I'm talking about today. And of course, you're always welcome to call me, contact me. We can talk offline to discuss any particular area with greater emphasis. And I definitely want you to ask me questions. I don't expect this to be familiar to you. But then again, I've designed this to where it's not for your biology majors or anything like that, but it's more for the person who wants to have somewhat of a better understanding of the neuroscience, the neuro components that cause a person to behave the way they behave. Okay. So, with that said, let's go and get started. The first thing we're going to talk about, and I'll spend a good 10, 15 minutes talking about, is the cell membrane. Well, more specifically, it's called the cell plasma membrane. Look at it as let me show you a picture of it. The cell membrane is this blue envelope that you see around surrounding the organelles that we talked about last week. Look at it as the shell. They call it the soma, the covering of the soma. And as I mentioned last week, it is porous. The cell plasma membrane is meant to introduce and excrete elements into and out of the soma or in and out of the cell, the neuron. Okay. And and that's what we're going to be talking about tonight. Talking about that transporting of in and out. Now, there's something else I'm going to cover next Tuesday. We call that cell signaling, which also talks about things that move in and out of the cell. But it's a little bit more specific. It's going to sound very familiar to a whole bunch of you because we're going to be talking about some neurotransmitters as well, which is very important for counseling. All right, but we'll get to that next week. So right now we're talking about a different type of cell transport. We're talking about the electrical conduction. What happens when the soma or these particular areas of your brain, your endoplasmic reticulum, your Golgi apparatus, your smooth endoplasmic reticulum, what the mitochondria does to generate the energy, things of that sort. So, what's the final output? What's the outcome? you know, we studied last week that the ribosomes, those specks that you see on the nucleus, on the rough endoplasmic reticulum, and really even floating around inside the cytosol, which is the gel structure that talked about last week. These are items that the ribosomes are what come from the DNA to help produce proteins. Protein structure is very, very important for neurotransmitters, electrical conduction, when cells move in and out, and what happens to the membrane when it expands and it allows things to become more porous. Proteins are very, very important, okay, for structure. So, with that said, let's talk a little bit more about the cell membrane. So, in order for things to move in and out, we have to have a better understanding of the cell membrane. It is made up of fat, or what we call lipids. Okay, it's what we call phospholipids. And if you can follow me, well, you're not gonna be able to follow me, but yeah, you will because I'm recording. If you look at my mouse here, you'll see on each end the tan color over here that is inside the cell. The gray area is outside the cell. Okay, so as you know, if you notice inside the cell, you'll see a head right here. On the outside, you see another head, and you see these two little stems that I've encapsulated, that I've drawn for you here. You'll see a head that has two legs attached to it. Okay, the heads love water. Okay, the poles, or what's called polar tails, do not like water. So anytime water molecules try to introduce themselves, or elements, or solutes that have some form of water in them, it's very difficult for them to try to go. They can probably go through the head, but they can't go through those poles. The pole will stop them. It'll prevent them from actually going through, and preventing them from going inside the cell. Sometimes, or not sometimes, these blue pictures that you see over here, one on each side, these are channels. Certain water containing solutes, or substances have specific channels that help move these extracellular, or another word for a definition for extracellular means substances or elements or solution or solutes that are outside the cell. Extracellular. Intracellular are items that are inside the cell. So for items to move from the outside of the cell, the extracellular elements to move into the cell, into the soma, into the cytosol, it's these channels are very specific. Sometimes you'll have glucose going through it. Sometimes you'll have sodium going through it. Sometimes you'll have potassium. They're very specific. Okay, and of course you're going to have these channels throughout your entire membrane. Okay, and that's how stuff gets in and out of the cell. That's how stuff gets transported into the cell and outside the cell. So let's take an example. Let's say, for example, you eat a certain item. Let's say banana. You eat a banana. Well, banana is very rich with potassium, K. If you look at the periodic table, K is potassium. Potassium is very important because it does something to trigger activity inside your cells. But of course, when you're eating something, your intestines will your stomach, and then your intestines will digest the banana. In turn, the potassium that's inside the banana will eventually be broken down to a certain solid sized particle that it will find its way into your bloodstream. And we've learned from last week that blood is nothing more than just a taxi cab taking all these nutrients, all these elements from point A to point B, and that's all the blood does. Okay, so let's say the potassium's hanging out inside the blood, and it's on its way to a certain part of your brain that's responsible for you moving your hands, motor type of cells. Okay, uh, but of course it needs potassium. So when you eat the banana and it gets broken down, digested, then after it gets digested, then the potassium finds its way into the blood and into the cell. Okay, the cell will finally look for, or tells you the potassium will look for a channel that is specific for potassium entry, and it just has a way of doing that. I mean, that's pretty. I mean, that's amazing that a potassium. I mean, you think of all the elements that are in your body. Channels know when potassium is there, and it finds its way to that channel, and it makes its way inside the cell. What does the cell do with it? Well, it goes into all these organelles that we talked about last week, and I'm not going to talk about it today because we already exhausted that. However, I want you to know that the potassium finally makes its way, and then your cell begins to fire the way it needs to fire, and you start getting contractions, which is fantastic, right? Well, let's take glucose. Let me give you one more example. Let's say a person has diabetes. Okay, person has diabetes. What that means is that glucose or sugar comes by what you eat, by what you drink, and it finally makes its way, is broken down by your intestines, your digestive system, and all kinds of other stuff. Okay, and finally, the sugar, or in this case, glucose, makes its way into your blood. And what was blood for? What does blood do? What's its main responsibility? To take these elements from point A to point B. It's a taxi cab. Okay, so let's just say it's taking the glucose from point A to point B, and it finally makes its way to the blood vessels around your brain, the capillaries and the arterioles and centrioles and all that. Okay, it finally makes its way to your brain cells, and it comes in contact with a neuron that is responsible for thinking, for being able to interpret something, a situation, a problem. Okay, and so the glucose, in order for that cell to function, remember, you're going to be thinking, you're going to be problem solving, critically thinking. In order for you to do that, the cell has to be fully functional. Okay, but how is it going to do that? It requires energy. We talked about that, where everything has to be active, but it takes energy. It takes adenosine triphosphate, ATP, to be able to move these elements and produce and manufacture proteins and process lipids and things of that sort. Okay, so where does it get this energy from? Well, the glucose finds its way close to the cell membrane. It finds a channel where the protein can go through. It makes its way into the particular blue channel, and it makes its way inside the cell and into the cytosol. And from the cytosol, what is the glucose attracted to immediately? Your mitochondria. And what does the mitochondria do? It takes sugar and the oxygen molecules. You see, oxygen doesn't have to go through a channel because it's so small and it's not polar. So it can actually go through the polar membrane here. So when sugar and oxygen make their way inside the cell and into the mitochondria, it makes adenosine triphosphate. It makes ATP. In turn, you create energy, and then your cells around the area of the brain that's responsible for you thinking critically can actually help you to think critically. But what happens when a person has diabetes? Let's say diabetes 1, where the pancreas does not produce the insulin. Or let's say you have diabetes 2, where you're just not producing enough insulin. Okay? Regardless of whether it's diabetes 1 or diabetes 2, what it amounts to is you're not producing the insulin required for you to get the glucose from outside the cell to inside the cell. Now, what am I saying? Insulin is used. Guess what area of the cell does the insulin impact the most? It immediately targets the cell plasma membrane, and guess what it opens up? It stimulates the channel that's responsible for taking glucose from outside the cell to inside the cell. But if you don't have the insulin, what's going to stimulate the glucose channel? Nothing. The channel does not open, and what happens? The glucose stays in the blood. So someone will prick their fingertip for blood to check their glucose level, and it's something like 400, 500. Your normal should be around 120, 130, right? But let's say it's 400, 500. Like, what the heck? Why do you have so much glucose in your blood? You should not have that much glucose in your blood. That tells me that the person's getting into the taxi cab, and another person's getting into a taxi cab, and another person's getting into a taxi cab. No one's getting off. After a while, it's going to be crowded inside that taxi cab. Well, that's exactly what's happening inside the blood. Glucose does not get off, and in turn, glucose never makes its way inside the cell. And let's just say that part of the brain is responsible for critical thinking. I don't know about you, but I've been very fortunate to work in the medical field, and I've seen somebody who's having diabetic shock. It's like, they're dazed. They're looking at you, but they got this blank stare because they can't process thought. It is because the cells that are responsible for that can't function because they're not getting the glucose, right? So immediately someone will give them a candy bar. Why? That's a lot of sugar. And that sugar says, well, let's put a bigger demand on your pancreas to release more insulin, and maybe then they'll start releasing some insulin when they start seeing the overload. Because remember, insulin does not get activated to be released until there's the presence of sugar. So that's why a person they'll give them a candy bar, and automatically all of a sudden they come back too. It's because you've forced the pancreas to release more insulin, allowing the glucose to go from inside the blood to, and the insulin activates the channel, the glucose channel. It opens up, glucose goes inside the cell, and then now the cell can function the way it needs to, and now the person is no longer in that state of that blank state. So what makes it important is sometimes you have elements that go through the cell membrane by way of channels, specific channels, and we're gonna learn 5 tonight, okay? And and that is the main objective for tonight, is for you to understand that we're trying to get elements from outside the cell to inside the cell. That's it. to activate these organelles to do what they're good at doing. All right. So the first thing, I'm going to show you some pictures here really quick. This is another example. This is another example of a cell membrane. You see the heads and you see the poles, and you see different types of channels. Okay, you have channels for cholesterol. You have channels for lipids, protein channels. You have all kinds of different channels. You even have glycoprotein, like what I was talking about right now. Certain channels for glucose and things of that sort. This is just another picture of, again, the cell plasma membrane with all these different types of channels inside the cell, outside the cell, extracellular, intracellular. Okay. So the first thing we're going to talk about is the two types of transport. You have passive transport and then you have active transport. To put it in simple terms, passive transport does not require ATP, or in other words, it does not require energy. It's just a natural flow. It's just a natural process of what your cells, extracellular, intracellular things do. Okay, and under passive transport, which does not require energy, you have simple diffusion. Then you also have facilitated diffusion, and then you also have channel mediated. Uh, I'm sorry, carrier mediated protein diffusion. Okay, so let's talk about the first one, simple diffusion. Basically, simple diffusion is just when you have things that are moving in. Let me show you the right here. Here's a picture of it. Simple diffusion. If you can look at this black arrow, things don't need a channel. They just make their way through the phospholipids that are aggregated together, and basically like oxygen, like you heard me say, carbon dioxide. What's carbon dioxide? After your cells finish producing the energy by getting oxygen and glucose together in the mitochondria, what's the waste product? Carbon dioxide. So carbon dioxide has to find its way outside the cell through the membrane and into your blood so you can breathe it out. Okay? So under simple diffusion, you have items that do not need channels. These blue elements that I have here, they can actually just penetrate the cell membrane. Okay? So CO2, O2 are gases. They're not polar. They don't have poles. They just simply have the heads, which is why they're able to go through any area of the membrane. They can just go right through it. Okay, it's such a small molecule, which is one oxygen molecule atom and two hydrogen atoms, O2. It's so small that it actually can penetrate the membrane at any point. Okay, and that's facilitated diffusion. And what's going to happen? With facilitated diffusion, you have wherever it goes from the place of where you have more of to the place where you have the least of. Okay, so you have an abundance amount of oxygen outside your cell. In this case, the blood. I should have drawn this red because it's really not gray. It's really where your blood is. What's going to happen is if your cells don't have that much oxygen, like in this case, you only have three molecules down here out inside the cell. Over here, you got what? 2, 4, 6, 8, 10, 12, 14 molecules of oxygen. So it immediately, the natural propensity to go to a place of lesser quantity to a place of to a place of more quantity to a place of least quantity is going to trigger this process. It wants to even out. It wants to spread the wealth. It wants to make things even. Okay, that's in simple diffusion. So this is a really good picture over here on this other side, on the right side, under figure 123, you'll see it, where things have evened out. And once they've evened out, the diffusion process stops. Of course, because it's a human body, there's never a point where you have complete equilibrium. In other words, where it's even and then just there, static. So basically, you have these smaller particles like gases that are constantly moving in and out of the cell. Every time you take a breath, when you inhale, you're bringing in oxygen molecules. When you exhale, you're exhaling carbon dioxide. So to say that you don't that it's reached equilibrium and you no longer need to diffuse is like me telling you stop breathing. Quit getting oxygen molecules in. Quit getting trying to get carbon dioxide out of your cells. So simple diffusion, it's always trying to balance things out, but it never reaches that. Okay. Let's see. Let's go to the next one. Is the facilitated diffusion. Okay. So Facilitated diffusion, again, is passive. It does not require energy. It has what we call an ion channel. Okay. For example, like sugar, glucose, what I just mentioned earlier. You know what? Let me talk about that in a minute. Okay. Let me talk about it more from an ion point of view. Ion point of view. Okay. Sometimes you have specific elements or ions that are very important for contraction, for generating energy, stimuli of some sort. You need certain ions to go from outside the cell to inside the cell. So you have these channels that they go through. They just naturally go through. Now, I'm going to show you another picture, which is very important because you have two types of these facilitated diffusion channels. Okay, you have the called channel mediated gate channel or a leak channel. Okay, let me show you a picture of both. So under facilitated diffusion, you have a gated channel or you have a leak channel. The leak channel just simply means there's a leak. It's like a faucet leak. It's always open. The channel is always open, but it is very specific to the ion. Just because it's open does not mean that any type of other ion can go through it. It is very ion specific. It remains open, a leak channel. Okay. The gated channel, it opens up just like the leak channel, but then it closes. But it's only sensitive to that particular ion. It does not require energy, it just does it naturally. Okay. And then we have the carrier mediate protein. And that's why I wanted to wait on the glucose because this is predominantly the most popular or the most prevalent diffusion process that happens. Again, what is passive diffusion? Does not require energy. You have a specific channel that is specific to glucose, for example, or proteins. Okay. So in this case, you have glucose that it's actually migrating from outside the cell to inside the cell. The funny thing about a carrier protein change, it changes its shape. Look at it as like if you've ever seen on YouTube or on some kind of TV channel special program or what it looks like when you're swallowing water, you'll see the peristalsis where you're swallowing, and it's like it's changing your esophagus changes shape because it's actually like contracting, opening and closing, opening and closing, opening and closing. That's the same thing that happens with a carrier protein or a carrier modified protein. Okay, and actually these channels. Well, I'm sorry. One thing I forgot to say is that these channels they're made up of proteins. Okay, so that's why it's called a carrier mediated protein. So the protein begins to change its shape, but the objective is to squeeze in and to squeeze in the glucose molecule. Okay? So those are the three types of passive diffusion. All right, and that's another example. And then let me get to active transport because I think we're about to go through a storm, and I'm afraid that I might get knocked out. So let me try to hurry things up a little bit. Okay, active transport, which is really the last thing we'll talk about. Active transport means just that it's active and it needs energy. It needs ATP. Now, let me say this first before I explain the active transport. Okay, and I'm going to talk about two. One, I'm going to talk about one today, but the other one I will talk about it specifically. I included it in a chapter in my book on cell signaling. Okay, that's another active transport. Okay, however, I only want to talk about a different type of transport we call active transport. Right. So let me show you a picture because I want to show you something. Okay, it's a channel. It's a protein channel, just like the ones you just learned under passive diffusion. Okay, but the only difference is if you notice this picture over here, you have an ATP. ATP means adenosine, which is very natural. It's a very natural element inside your body that you create, not because of your DNA. But you have triphosphate. That means you have three phosphate molecules. And when you put those together inside the mitochondria makes ATP. It gets the adenosine and it makes the three phosphates using oxygen and glucose and some other things that go on. Okay. And what happens, it produces an ATP, adenosine 3 phosphate or adenosine triphosphate. Now, what happens is that ATP is inside your cell, it's intracellular. So what happens as soon as that ATP gets close, or let's just say the channel puts a demand on your mitochondria to send over some ATP because it's about to do something here. it requires the ATP to get real close to it. As soon as it does that, one of the phosphates is released into the protein channel, which in turn, after it releases it, that phosphate actually stimulates the activity of that channel. And you look at it as, and it has this movement. It's first like this. It's like a tent, a V, an upside down V. And what happens? Let's say you have a lot of potassium inside your cell, which is very, very common. You typically have so much more sodium outside the cell than you do inside the cell. And you have a lot more potassium inside the cell than you do outside the cell. So what happens is when sodium and potassium go in and out of the cell contracts. It creates an energy. Okay, and that's what we mean by stimulating. Okay, so what the way things get stimulated is the ATP, once it gets close to the protein channel, it releases the phosphate and it activates the channel, the protein channel, and turn three potassiums link up to those three specific target areas. And then what happens, it changes motion, it changes from an upside down V, and that phosphate actually causes it to turn into a V. Well, at that point, if you look at this picture, picture C, you see how it changed? Compare the picture A to the picture F. I'm sorry, A to C. See how you have an upside down V, and over here you have a V. Well, what happens when it's an upside down V? This phosphate that's here, just go to picture B. The phosphate is now detached from the ATP. The ATP becomes adenosine diphosphate. Di meaning two phosphates. Okay, so the protein stimulates this protein channel, in turn converts it, and now those proteins are released to outside the cell. So these three phosphate. I'm sorry, sodium. I'm sorry. I'm showing you. I'm talking about potassium, but I'm actually showing you sodium. It doesn't matter. Whatever the ion is. In this case, sodium. There's three sodiums that now make their way out. See how they got brought in here? And A, it was strengthened here in B, and look what happens in C. That phosphate caused that protein channel to change posture, releasing the sodium to the outside of the cell, and guess what came in? Two potassiums, and they linked up on this other side. See how that happened? Well, guess what happens? The phosphate wore off. Now what happens? The protein channel went back to its natural shape, the upside down V, and look what happens to the potassium. Potassium comes inside the cell. So that's all you have. You have a bridge opening and closing. And as it's opening, it's releasing one element, and then it's closing, it releases the other element. And that's constantly happening. Okay. So if you've ever wondered, you've heard me talk about how sodium and potassium comes together, and because again, you have both of those electrolytes inside your cell and outside your cell. But as I mentioned, you have a lot more sodium outside your cell than inside your cell, and potassium, you have a lot more potassium inside your cell than you do outside. So when the ATP causes this reaction, and your cell membrane is channel, in this case, the cell membrane is activated by way of your channel proteins, you have an exchange. Sodium comes in, potassium goes out, and that causes a contraction, and now your cell is responsible for whatever it's assigned to do. And as I mentioned in the first class session, every cell has a purpose. So if this particular cell, let's just take for example this cell that I'm showing you right now. Let's say it's responsible for being scared, being fearful in your limbic system. Your limbic system has been activated, and all of a sudden you're scared. Okay. Well, what if there was a bear in front of you and these channels weren't working? So you can't have the sodium potassium going inside the cell. Therefore, that particular